Enzymes. A catalyst is a substance that increases the rate of a chemical reaction and is not changed by the reaction. Enzymes as proteins that function as a biological catalyst. Let's understand, why are enzymes important? Enzymes are necessary to all living organisms as they maintain reaction speeds of all metabolic reactions at a rate that can sustain life. Let's see, how do enzymes work? Enzymes are specific to one specific substance called a substrate. The substrate is broken down as the active site, where enzyme and substrate bind. The shape of the active site is the complementary shape of the 3D shape. This is known as the lock and key hypothesis. When the substrate moves into the enzyme's active site, they became known as the enzyme's substrate complex. After the reaction has occurred, the products leave the enzyme's active site as they no longer fit, and it is free to take up another substrate. Enzymes and substrates randomly move about in solution. A product forms from the substrate, which is then released from the active site. The enzyme is unchanged and will go on to catalyze further reactions. Now see the effect of temperature on enzyme functions. Enzymes are proteins and have a specific shape, held in place by bonds. This is very important for the active site area, as it ensures the substrate will fit into the active site. Enzymes work fast at their optimum temperature. For most enzymes, it is 37 degrees. If enzymes are exposed to very high temperature, it causes the bonds in the enzyme to break. This is known as denaturation, SARS, irreversible process. Secondly, see the effect of pH of enzyme functions. The optimum pH for most enzymes is pH 7. If pH is too high or low, the bonds that hold the amino acid chain together to make up the protein can be destroyed. This will change the shape of the active site, so the substrate can no longer fit into it. Moving too far away from optimum pH will cause the enzyme to denature, and activity will stop. Lastly, the enzyme investigations. Amylase is an enzyme that digests starch, polysaccharide of glucose, into maltose, a disaccharide of glucose. Starch can be tested for easily using iodine solution. Testing amylase.